Right, hello. So, we have gotten some big news, and by that I mean me. I now know when all of my exams are for May, which is kind of worrying because that means that now, let's just say that it's March, right? We now have exactly 2 months and 2 weeks before AS levels, which is terrifying. The worst bit, I would say, is the fact I actually have 2 exams on one day. So I'll be doing a biology exam and a core math exam on the same day, which sucks. That means that I'm probably going to get disadvantaged in some way there. I hope that I'm able to get like a break and stuff in between, because otherwise I feel like that's just awful, sitting in an exam hall for like 4.5 hours. But anyways, what this means is we now know how long we have. The clock is now ticking. 2 months, 2 weeks. That's all we have to get the most out of my revision. So, I guess there's no time like the present. Today, I'm actually going to be revising what I would consider the most difficult biology topic so far that we've done, and it's the DNA topic. Now, instead of what I was originally going to do, where I was just going to do a mind map, what I think would be interesting is if we went through the teacher's PowerPoint, wrote some bullet point notes, drew some diagrams, and then we just kind of see what happens. We maybe do some exam questions at the end. Because I honestly don't know much about this topic. Obviously, we've done lessons on it, but I was off during 2 of them, which means I am very much behind on this topic. So I'm going to pull out my computer. And yeah, this is probably, again, I would argue the most difficult topic in biology is DNA, purely because not only do you need to know so many definitions, but you also just need to know a lot of key information about processes. Now, what I'll be doing tomorrow is just some generic cell biology revision. Going through the microscope topic, the cell fractionation topic, things like that. So today's all about DNA. So, here we go. DNA and chromosomes. This is the first topic. And we already know some information, so I guess we should start right away by plotting down what we already know. So I know that there is a difference between prokaryotic and eukaryotic DNA. The difference between prokaryote and eukaryote DNA is eukaryotes, the DNA is wound through, I think they're called histones, which are, I believe are like basically protein kind of ball things which get wound around and they kind of add support to the structure of the DNA. And obviously prokaryote does not have histones. So, DNA is the same in all organisms, the structure, but the way it's stored is different. So, eukaryotic cells, yes, the DNA is linear. But in prokaryotic, the DNA is circular, aka plasmids. Or it's just kind of free-floating too. Made up of one long molecule. DNA molecule is long and wound up. So yeah, histones is what it's kind of wound up into. The DNA molecule is wound around, they're proteins called histones. Histone proteins help support the DNA. The DNA and protein is tightly coiled. So it's coiled into, obviously, I'll just draw like this. chromosomes. But I don't believe chromosomes exist, and chromosomes are found in the nuclei. Now, prokaryotic cells, mitochondria and chloroplasts have DNA similar to prokaryotic DNA. It's short and circular, and there are no histones. So yeah, that's another thing. Prokaryotic DNA is shorter. So, the first question kind of here that we have is, recall the structure of eukaryotic DNA. So the DNA itself is in a double helix. It's wound around proteins called histones. I don't know, I guess that would just be the DNA. That's the double helix. Sorry, I'm talking to myself here. That's obviously the double helix. That one there. That would be DNA. That would be the histones. That would be a chromosome. That would be, what are these? Just getting wound up? Yeah, wound up around the histone proteins and coiled up. So two different things there. The first is that it's wound around the histones. The second bit is that it's then coiled up. So it's wound and coiled. Interesting. Now, prokaryotic cells, we know a lot about this. The pili of little, I don't know what you would call them, kind of like the little hairs. The flagella, the tail, basically. The plasmids, circular DNA. The slime capsule for protection. It has a cell wall made out of murine. It has, obviously, cell membrane. Ribosomes. I think they're 70S ribosomes, not the 80S ones. Obviously, free-floating DNA. Prokaryotic cells are smaller. DNA is not contained in a nucleus. So, obviously, they're not contained in the nucleus. Prokaryotic organisms that consist of a single prokaryotic cell. Okay. So, DNA is attached to the cell membrane. So it's attached to the membrane. Which I didn't know. So it's attached to specific attachment sites. Okay, that's that one word. So prokaryotes carry DNA. The DNA molecules are short and circular. DNA isn't obviously wound through the histones. It condenses. It condenses and supercoils into obviously the membrane. That's pretty cool. So it attaches to the cell membrane. And I think that's via the binding proteins. I'm going to go buy some new whiteboard pens because these are not going to last me another couple of months. I know for a fact. And I need these next year too. It's an actual miracle that these have lasted as long as they have. You know what I'll do? I'm not free to... Thursday. I'm free for an hour and 45 on Thursday. I will go buy some whiteboard pens. So, just like eukaryotes, obviously, the prokaryotic DNA is a double helix, okay? But instead of it being linear DNA, it's circular DNA. It's not wound around the proteins called histones. Instead, it supercoils, it condenses so that it can fit inside the prokaryote, and it's attached to the membrane via attachment proteins. All good. So they all have cell membranes, they all have cytoplasm. Thing is, eukaryotic cells are more complex. Yeah, we know this, we know. Okay, describe the structure of eukaryotic chromosomes. Again, we know this. Eukaryotic chromosomes, so the DNA is initially a double helix, which is wound around proteins called histones, which provide additional support to the structure. This structure is then, coiled up into a chromosome. I don't know what else you would put there, really. They are wound up. I don't know how many marks this is. Describe the structure. So we've talked about it being linear DNA. We've talked about histones. Talked about it being coiled up. I guess you could just emphasize that being linear. Contain linear DNA that exists as chromosomes. It's one long molecule. The DNA molecule is long and wound up, wound around proteins, which support the DNA. It's tightly coiled, and and they're found in the nucleus. Okay, I missed two things there, I think. And prokaryotic, obviously, does not... describe how it differs from DNA in eukaryotes. So in prokaryotes, the DNA is circular. It is not found in nuclei. Instead, it is attached to the cell membrane via attachment proteins. The DNA supercoils, as there are no histones for it to, wind around. I guess that would be it. It isn't wound around histones. Oh, and it's shorter. It's shorter DNA. I forgot to mention that. Okay, genome versus proteome. What is the difference? I don't know, so let's find out. A genome... Oh, hold on. This is not genome versus... genome... So a genome is the entire set of genes in a... So, like the Human Genome Project. Okay, that makes sense. Proteomes are a set of proteins. Okay, that honestly makes sense. I would argue that that's okay. Genome is just the entire set of genes. The proteome is the entire set of proteins that a cell produces. Cool. So what is a gene specifically? This is a very important definition. I'm going to make a guess beforehand, because again, I don't know this topic. I'm going to assume that I'm pretty sure it's a part of a DNA. Like, part of DNA... which codes for a specific polypeptide. A gene is a section of DNA located at a particular site of a DNA molecule. So a gene is a base sequence of DNA that codes for the amino acid sequence of a polypeptide and a functional RNA. There's a lot of information here, but it feels like not much of it's going on. A gene is a section of DNA. So a gene is a base sequence. Let's break that down first. Base sequence, so obviously, the different types of bases obviously code for different things. That makes sense. It's a base sequence of DNA that codes for, I believe it's a polypeptide. Oh my god, it's getting really bright and dark all of a sudden. The amino acid sequence of a polypeptide. So that codes for a polypeptide. And a functional RNA. So two things that it codes for. The polypeptide of a functional RNA. So I'm going to underline those. So it codes for a polypeptide and functional RNA. That is okay. I would argue that's okay. It's just a little sequence of a gene, a little part of a gene. A locus is a position of a chromosome occupied by a gene. So I'm just going to draw the chromosome here. So we're saying that a locus is obviously a specific part of a gene. Well, not a gene. Oh my god, it's so dark all of a sudden, what the fuck? It's a specific part of a chromosome where the gene is contained. Cool. Okay, this is where we get into the more complex territory. So yeah, a gene is a sequence of DNA that codes for a polypeptide or functional RNA. I should probably change that to all. So, I guess this is a summary, a rough summary here. Where is the sun gone? So, to make the polypeptide, firstly the DNA is copied into messenger RNA. It is the order of bases in a gene that determines the order of amino acids. Each amino acid is coded by a sequence of 3 bases. So, obviously, the different bases code for different amino acids. So each amino acid is coded for by a sequence of 3 bases called a triplet. So I guess. Okay, hold on. Before we continue, alleles, I'm pretty sure, are a different form of the same gene. Genes exist in more than one form, alleles. The order of bases in an allele, I'm going to put this over here. So an allele, the order of bases in each allele is different and code for different versions of the same polypeptide. So it's basically different a different form of the same gene. So, in other words, yeah, that does actually make sense. It's a different form of the same gene. The order of bases is different, which means they code for different versions of the same thing. Next, the homologous chromosomes. So, homologous chromosomes is where one chromosome is inherited from the male, one is inherited from the male. Sorry, one inherited from the male, one inherited from the female. That makes sense. Finally is the non-coding DNA, which we already know, exons and introns, but we'll very quickly... Extrons are basically pretty sure non-coding DNA can be found in genes as introns. Non-coding DNA, which is removed so during transcription, the whole thing is transcribed, including the introns. But before it leaves the nucleus, that's when the introns are removed. So introns removed. Alright, that honestly makes sense. Okay. So far all of this makes sense. I understand all of this. The next part, I feel like, is a massive jump up in difficulty. So let's very quickly make sure we actually know what we're waffling on about here. The first one I don't get. Draw the homologous chromosome. How would you do that? Oh, hold on. I didn't get the definition. That's why I'm confused. Homologous is just a pair of matching chromosomes. It's a pair of matching chromosomes. That makes sense. I'm going to move this back a little so you guys can see. Oh, that's fine. Okay, so we can draw the first one. The diagram below shows a sequence of a short stretch of DNA. Write down the base sequence that actually determines the order of amino acids. So you would just write down everything that isn't an intron. How many bases long is the region of multiple repeats? 1, 2, 3, 1, 2, 3, 1, 2. Okay, hold on, that doesn't make sense. That's confusing. And there's no answers. That bit doesn't make sense. How many bases long is the region of... Or is it on about that region? No, it's not about that region. 1, 2, 3, 1, 2, 3. No, I don't understand that one. How long is a region of multiple repeats? That doesn't make sense. What? None of them are 3 long, unless I'm misinterpreting. It is the order of bases in a gene that determines the order of amino acids. Yeah. I don't get it. I don't get that. That is a weirdly worded question there. It doesn't really make sense. Let's skip that. Write down the base sequence is repeated in a Again, these questions are kind of goofy here. It doesn't really make sense. I'm going to skip that. What is a gene? Obviously, it's a part of a... Oh my god, how have I forgotten this already? It's a part... Oh, no. It's a base sequence of DNA that codes for a polypeptide or RNA. I think. Yeah, a base sequence of DNA that codes for a polypeptide or functional RNA. There we go. How many DNA bases code for one amino acid? 3 bases is one amino acid. A triplet. What is a cell's genome? A genome is all of the genes, basically. An entire set of genes. Sorry, I've said it in a really informal way. A proteome is all of the proteins produced by a cell. Oh, sorry, the entire set of proteins produced by a cell. Name 2 types of non-coding DNA. I guess just introns. Yeah. What's it asking there? I'm confused. Introns, or I guess you could say repeating the multiple repeat regions. Name the sections within genes that code for amino acids, the extrons. What is an allele? So it's a gene which I guess is... man, I literally just wrote down the definition and I've already forgot. This is how weird this topic is for me. I don't remember any of the words or the definitions. it's the same the gene. How do I explain this? It's the same gene. Oh fuck! A different form of the same gene. I knew that, but I didn't know how to explain it. Fuck. Okay. Alleles for the same characteristic can be found at a particular fixed region on a chromosome. What is the name given? A locus. Okay, we're done. We're done with that lesson. That's only the introductory lesson. There is still the transcription and translation lesson. So, we know now. An allele is a different form of the same gene. A gene is a base sequence of DNA which codes for a specific polypeptide or functional RNA. We know the difference between prokaryotic and eukaryotic DNAs. Prokaryotic, it's circular DNA, which is shorter, with no histones, and it supercoils in the cell membrane via attachment proteins. Eukaryotic has histones, it tightly coils into chromosomes, it winds around the histones, and it's linear DNA, which is longer. So, here are the key ideas. Protein synthesis takes place in two stages. Transcription, where one gene on the DNA is copied into messenger RNA, and then translation, where this messenger RNA template has moved to the cytoplasm and it joins with a ribosome. From there, corresponding transfer RNA molecules bring the specific amino acid that the codon codes for. Honestly, reading that has kind of made it a little easier. So it goes like this. So if you want to code for, I don't know, a specific hair colour gene, let's go back to primary school here. Then, first of all, the gene, I guess, would be identified in the DNA. A copy would be made of that specific gene through transcription, which will then go out of the nucleus in the cytoplasm. There it joins with a ribosome. Then transfer RNA actually transfers the specific amino acids that it codes for. That kind of makes sense. So, that's the first stage, transcription. So in biology, transcription is a process of copying out the DNA sequence of a gene. The goal of transcription is to make an RNA copy of a gene's DNA sequence. So RNA is a smaller part of the DNA sequence, basically. It's a small part of DNA. It's a non-double helix, it's a singular helix. It's smaller and it has a different base. Instead of ATCG, it's AUCG. So already we can write down that it basically makes an RNA copy of is it a gene, I guess? Of a gene's DNA sequence. Which makes sense, right? In fact, I'm even going to draw like a kind of double helix here to kind of highlight this. So let's say we wanted to identify, and obviously this is such a crude drawing, but I'm doing this on purpose here. Let's say we wanted that specific purple gene there. Obviously, that looks like a base from... But it'll make sense in a little. So yeah, DNA contains a lot of genes, so it's too big to just get the whole DNA itself, so you have to make a smaller copy. That makes sense. The RNA copy carries the information needed to build the polypeptide. So how does this work? How does transcription work? So number one, the helix unwinds to expose the bases. So number one, it unwinds. And then we're going to put the main point here. So it unwinds to expose bases. So it unwinds to expose the thing. Now this is similar to actually DNA replication, where it unwinds, the free-floating DNA bases bind and it replicates the DNA. Instead of this, only a singular chain of DNA acts as a template, and that's why RNA is not a double helix. There you go. I just came up with it on my own and it works. So only one chain. So it exposes the bases. So only one chain acts as a template. So like that, basically. That's cool. That's a cool drawing. There. It looks like a comb. See, we're actually doing quite well here. We seem to be getting the gist here, guys. Eventually. So, only one of the actual DNA chains acts as the template. Similar to DNA replication, it unzips, and it's catalyzed by DNA helicase. So I'm going to write that there. So yeah, that's a cool drawing, which makes sense. So, 3 mRNA bases, so unlike before, in fact, we could simplify this even further. We are writing a little bit too much here. Unwinds via... I don't know, actually, because I just figured unwinding actually does make sense. No, sorry, I have tried to oversimplify this to the point where it's wrong. Let me go back now. So, when it unwinds, this is what it ends up like. It ends up just as 2 individual lines. Instead of it being a coil, it's 2 straight lines. That makes sense. Got it. Similar to DNA replication, it unzips. So this time, so now it's 2 straight lines. These lines, which are still kind of joined. So it kind of goes like this. Then it goes like that because it's unwinding. And now it's going like that. Or I guess like that. That makes sense. That genuinely does make sense. Let's even... You know what? I'm taking my sweet time here because I'm trying to make this as simple for you guys and as me as possible. Let's draw this, like, really accurately. So that's obviously the DNA. So it unzips. Oh, sorry, it unwinds. There we go. Then it unzips, which is where you then have, again, I don't know how I'm really going to draw this. There. That genuinely makes sense. Unzips. This is only one part of a topic, but I'm trying to... Well, this is only half of the actual thing, but I'm trying to make this all make sense. So it unzips via DNA helicase. Then the three mRNA bases align. Align with the one chain. So there you go. So the three bases now align with the one chain. Not both, because it's not replicating the DNA. It's only making a copy, a singular chain copy. Then, RNA polymerase bonds them together. So now, I guess we're going to draw this with a different colour. RNA polymerase joins together the nucleotides by creating the phosphodiester bonds. So now it's called pre-mRNA. Oh, pre-mRNA. Okay. Hold on, I've kind of drawn this diagram wrong here. You see, what actually happens, and this actually makes a lot more sense, is... Okay. So, what actually happens is it's a singular chain. Then these individual bases are the things that get joined. So that's via RNA polymerase. So RNA polymerase creates a separate chain, like so. And it does that while it's actually unzipped. So again, I've kind of drawn this diagram wrong in a way. What it really should be like is... Motherfucker. I get it now. I genuinely do get this now. This actually does make sense. It just took a lot of time. Because again, it's not making a copy of a whole thing. This makes sense. This is actually starting to click. This is actually starting to click now. I hope it's starting to click for you guys. So it's called pre-mRNA. So, pre-mRNA, then the pre-mRNA, the introns are removed, the extrons are kept. So, there you go. I'm genuinely happy with that. I think this genuinely makes a lot of sense. So firstly, if we do a little diagram, a hand gesture from before, the DNA actually unwinds, so now it's like that, but the hydrogen bonds are keeping it together. Then it unzips via DNA helicase, and now you're going to have to imagine like this pen is, I guess, the bases. The bases then, they basically take a screenshot. They basically go down one side of the DNA, they take a screenshot, and then they go out, basically. So all they're doing is these random ass bases are actually matching up, and that's when they're then joining up. This totally makes sense. No, I get this now. So, transcription. Let's do this without looking. So transcription. So firstly, the DNA unwinds. Then the hydrogen bonds are broken by DNA helicase, exposing both, DNA strands, but only one strand acts as the template. That's the important thing to remember. From there, the free-floating RNA bases will then align to their complementary base. They are then joined together by RNA synthase, which creates the phosphodiester bonds. So now you have this individual RNA strand, which is completely separate from the DNA. From there, it will exit the nuclei, but before it does that, it becomes pre-mRNA. As pre-mRNA, the introns are removed, the extrons are fused together, giving you a full RNA strand, which then exits the nuclei. That was a lot. So RNA polymerase attaches to the strand of DNA. The DNA strand separates, which exposes the nucleotides. Sorry, I should have said nuclei. Actually, no. That should be bases. The messenger RNA pairs with the DNA template. Nucleotides are added until the RNA polymerase releases it. That's cool. That's cool. This all makes sense. Now we move on to tRNA. This part I feel might be a little bit more challenging. So, I don't know actually. I don't know. This part I feel like could be easier. I feel like we got the hard part over and done with. Yeah. Oh. Okay, it turns out I got to go on a dog walk, but just as I was starting to get the hang of this, so I'm not going to right now. I'm going to go quickly message, and I will be 1 sec, and I'll be right back with you guys. We will get this topic done, because I'm getting the hang of it now. I'm getting the hang of this. Okay. Okay, where were we? The transfer RNA structure. This pen's actually surprisingly good. I'm surprised I haven't used this one that much. So, this is what the structure looks like. It's kind of like a big T shape. It's kind of like a T shape with a kind of little radio signal thing attached. So this is tRNA. So. Okay, great. We have plenty of time. So. I just like wipe my face and there's blue pen all over my computer now. What the fuck have I done? That's not coming off, is it? So, this itself is the tRNA molecule. Cool. Those are the... Okay. There is a little bit more we kind of missed here, so let's get this pen out. So there are actually bases here. This is like a weird mishmash of a DNA molecule. This looks really weird. And as you guessed, I mean, this bit's almost obvious, there are hydrogen bonds between the bases. There are hydrogen bonds between the bases. That makes sense. There are 3 amino acid... There's an amino acid attachment site. So I assume that's where the amino acids actually go to get made. Right here. Amino acid attachment. So, you could almost consider this thing like a factory, in a way. I mean, think about it this way. This whole sequence is like a factory. You almost get like your building schematic. But look, imagine it like this. Imagine like you're told to make like a specific, like, as catalogs, for example, where there's like thousands of like products in. Let's say you get asked to make specifically one of them. Well, firstly, you obviously have to go through, find the specific one you want to make. Instead of, ripping out the page, what you're going to do instead is you photocopy it. So firstly, you, I guess, unwind the book, find the right page, kind of unzip it, kind of almost rip it out to put it on the photocopier. You copy it, and then you kind of glue it back in the book. And then from there, you kind of maybe go with like a Sharpie, cross out any of the bits you don't need. That would be your pre-mRNA. Then from there, the tRNA is actually the thing you use to make it. So you get your schematic here, and then that's actually how you make it. That's actually a decent way of remembering it. If you can genuinely use the catalogue example, that would be sick. So yeah, that's the amino acid attachment site. This right here is the kind of key bit. And I'm gonna actually draw the DNA as I do usually. There we go. Okay, so right there is... So it has an anti-codon, that's what this is called, binding site. And that itself is the codon, so they join together. And when they join together, so it's a series of 3 bases which are complementary to the codon on the mRNA. Hold on. a series of free bases that are complementary to the codon on the mRNA, which codes for the amino acid the tRNA molecule carries. So each tRNA only codes for a very specific molecule. Yeah, it has a specific amino acid which codes for a specific anticodon. Okay. This is starting to get a tad complex here. So that's mRNA. So that contains a codon, and they obviously bind. So from here. Okay, now we've entered some complex territory. Translation. So, from here, the mRNA leaves the nucleus and it attaches to the ribosome. The ribosome attaches at the start codon, so basically the beginning. From there, the transfer RNA molecules float, and they bring a specific amino acid. So, in other words, okay, hold on. Let me try and comprehend this before reading it. So the RNA enters the ribosome. The transfer RNA molecules bring a specific amino acid, which are then held in place. The ribosome moves along the molecule, attaching another tRNA molecule. So in other words, the mRNA this bit kind of makes no sense. I'm going to be completely honest. This bit is kind of confusing. A ribosome binds with the messenger RNA. Transfer RNA has a complementary codon. So the tRNA and the mRNA combine together. The tRNA carries an amino acid. The ribosome then moves along and more amino acids get added. No, this makes complete sense. I got it. So, we have good old big ass ribosome here. I'm going to draw it a bit like a hamburger. Then we have our chain here. It's like a happy hamburger. You guys will get what I mean in a second. Okay, so the tRNA, which I'm just going to draw as like a little plus sign. The tRNA brings a specific amino acid. So in other words, the mRNA joins to ribosome. tRNA Okay, so this is what happens. So we'll go all the way back to the beginning because I need to go in about 10 minutes. So let's just go all the way back from the beginning. We'll use our hand example. So firstly, the DNA molecule unwinds. So it's like this. Then the DNA helicase breaks apart the hydrogen bonds between the bases, causing it to unzip. From there, an RNA, well, the free-floating RNA bases will basically create a copy of one strand of the DNA. So it'll create a copy of one strand because the complementary bases are aligned. Then RNA polymerase joins together the phosphodiester bonds. So imagine initially it's like a bunch of random shit. It kind of flies in, well, it kind of moves in, you know. It lines up, and that's when the RNA polymerase joins it together, so that now it's an exact copy. Alright, I guess let's actually do this the proper way. See, we have this here, we want to make a copy of it. Imagine these two things here are free-floating RNA bases, well, you know, the complementary bases. They line up at the specific parts, but they're not joined together, and that's when RNA polymerase joins it together. So now it's a copy. So then the copy goes, but before it can leave the nucleus, the introns get removed, the extrons are kept. Then it leaves the nucleus and goes to the ribosome. Now at the ribosome, the first three bases of a codon attach to a specific tRNA molecule with a complementary anticodon. So it's a bit like this kind of thing. They join together. So imagine this is the RNA, this is the the anticodon. So they've joined together, okay? And from there, imagine this little O shape here. This O shape represents an amino acid. So we have our RNA here in the ribosome, and this comes along, it brings an amino acid, and then it pisses off. That moves down, the next one. It's like a conveyor belt. So when mRNA moves along, a different codon is shown, which means a different tRNA then join. Okay, no. I'm explaining this badly, but I understand it. So mRNA comes into the ribosome, and the first three bases, the codon, are, I guess, shown. Or they attach, sorry. So the first codon attaches. From there, the tRNA molecule with the complementary, sorry, I'm butchering my words. The tRNA molecule with the complementary anticodon, that then joins up, they attach, and that tRNA molecule has actually been carrying the complementary amino acid. So what it does, well, it's, I guess, amino acid, like a little gift. And then it leaves the amino acid there, and as it moves along, new tRNA molecules attach to their complementary anticodon. So that eventually, it creates a chain of amino acids. It creates a little chain of amino acids because all three codons... Got it. I understand this topic very well now. I get it. This all makes sense. So mRNA joins to the ribosome. The first codon attaches. The tRNA molecule with a complementary anticodon brings its specific amino acid. The ribosome then moves along the mRNA strand, and every time it's basically... So, okay, hold on. Let's go all the way back to my catalogue example. Here, watch. I'll even go through the Liberty, guys, to make this look better. We're going to get out my old artwork folder. So look. So imagine, I don't know. Imagine this is the DNA. right? A DNA strand. Okay, well, first of all, imagine it's locked. Okay, so firstly we obviously need to unlock it, aka unwind the DNA. There we go. I kind of went a bit preemptive here. So now it's like this. Then obviously the DNA helicase will cause it to unwind. So now we have all of... Oh my god, I'm making such a mess here. Now we have all of these free floating... Oh, sorry. We have all this acting as a template. Again, we're going back to the catalogue example here. So it unzips. Then the free floating RNA comes in. Or I guess for our photocopy example, that would be, you're finding the right page. You then obviously create a copy of that page using a photocopier. I get it, but I'm kind of tired, so this is getting a bit hard to explain. But I hope you guys understand now. I hope you guys understand, because I do. And I am chuffed that I understand that, because that is a tricky-ass topic. I would argue that's the most difficult topic we've done yet in biology. I would argue that is the most difficult topic, and I can promise you guys that if you watch this stream through its entirety, I hope that you guys also understood it as well as I did. Because I was talking to Faith, actually, last night, and she was on about how she didn't... Well, I don't know, we oh, I flipped this folder up, shit. Great, guys, my flipping biology examples got ruined years of art. Okay, I'll deal with that in a bit. But I was talking to Faith, and we kind of got on the topic of biology, and I was talking about how this is, in my opinion, the most difficult topic. I think the thing is, I'm almost glad we're doing all the difficult stuff now, because it makes it a lot easier in the long term. Because imagine if we were doing the biological molecules topic at the end of year 13, we would be screwed. But the fact we're doing all the difficult stuff right at the beginning gets it over and done with, and that's what I'm happy with. So there you go, guys. So, let's do... Should we do... Because I have maybe 10 minutes. Let's go on this website. Let's do a quiz. If it'll let me... Yes, it will. Again, I kind of lost focus during the end there, but we know. Okay. So DNA and protein synthesis. This is what we're doing. Okay. Transcription and mRNA. What type of molecule is a transcription factor? A transcription factor? I don't know, actually. I don't get that one. What the hell are these? Epigenetic control of genes? Nope, I don't know that. Specific mRNA strands can be targeted for destruction. We have not done any of this. Okay, hold on. Is there an easier topic? Okay, 13 questions. Which term is a thread-like structure of nucleic acids and protein found in the nucleus of most living cells? That would be the chromosome. Yeah, because it's the proteins of the histones. What are the names of the proteins in which DNA is wrapped around? Histones. Which term is a section of DNA that codes for polypeptides or functional RNA and is located at a fixed position? Would that actually be a locus or a gene? Because a locus is a specific position. I'm going to say gene. But yeah, because the gene is located on the locus, yeah. Each human chromosome is comprised of one DNA molecule. No. They're comprised of two. Two strands of DNA. What is the end of a chromosome called? The end? It's not allele or locus. Telomere, maybe? I don't know. We haven't done that. What is the specific terminology for a position on a chromosome locus? DNA replication occurs during which phase? Interphase. What type of chromosomes are found in eukaryotic cells? Linear. What type of chromosomes are found in prokaryotic? Circular. Prokaryote and eukaryote DNA use the same bases. I want to say yeah. Prokaryotic DNA is not wrapped around histones. The extra molecules of DNA found in some prokaryotes are plasmids. The name of a pair of chromosomes are homologuses. DNA molecules have an overall negative charge. I don't know. Complete guess. I'm going to say... I want to say that might be true. Because amino acids do have a slightly different charge. You inherit your mitochondria DNA equally. Mitochondrial DNA is circular. Mitochondrial DNA is evidence for evolution. And chloroplast contains circular DNA, yes. %. The only ones I didn't get right were... Okay. Oh, yeah. No, I'm such an idiot. Of course, every human chromosome is made of one molecule. I'm an idiot. Sorry about that. You inherit your mitochondrial DNA from your mother. I actually didn't know that. And the last one I got wrong. Yes, I was going to put endosymbiosis, but I was like, hold on, that might be wrong. It wasn't. I'm happy. That is sick. Thanks, guys. Thanks, guys, for watching. Genuinely. We have just covered what I thought was the most difficult topic in biology. Now I think it's one of the easiest. It's still difficult, but so long as you kind of almost think of a factory example. Here's words you need to know. Unwind, unzips, DNA helicase, the free RNA bases, singular chain, RNA polymerase, pre-mRNA, introns, extrons, ribosomes, codons, anticodons, tRNA, amino acids, complementary, Fifteen words. The DNA unwinds, it unzips via DNA helicase, the free bases, the free RNA bases align with the one chain, the RNA polymerase. You know what's kind of mad? The fact that we spent 3 lessons doing this in class, I went through the whole of this topic and understood it better than the teacher taught us in an hour. I'm happy with that.